We're gonna get going in a couple of minutes. Just before we formally get started in today's class, I don't know if you are aware of it or not, but today is actually the International Women in STEM Day. So some of you might be wondering why support women in STEM. So here's the reason why. If you look at the makeup of our class, it's roughly 50-50 male to female, and yet if you look in academia, and particularly in the STEM fields, so research, teaching, most of the researchers tend to be male. So it's now reported, even though it's getting better, so it's roughly a 70-30 split. So 70% male, 30% female. And so really supporting women in STEM and giving you a clear path toward maybe thinking about a future career in research, in academia, in teaching, or in clinical practice, whatever it is actually really important to us because it should be representative of the general population, which is 50-50. And so that imbalance is something that is recognized and is something that we want to encourage being shifted more toward an equal balance for better representation. And so without detracting from the time that we're going to use in lecture today, we also just want to acknowledge that it's Women in STEM Day. And if you feel comfortable doing so, we're going to take a picture and upload it as a class for support of women in STEM. So if you can just come forward, we're going to take a group picture, and we have our TAs here as well. So if you guys also want to come forward, we're going to do it right before class. We still have about 2 minutes to go. And we'll try to take a really quick picture. We're going to just show that our class supports women in STEM and recognize that there's still some issues there. So if you are comfortable, come on forward and we'll take a quick picture. Yeah, so we're going to try to stand a little bit toward the back if you're okay with it.
Wow, this is such a different view of the class. Yeah, actually, it's so sweet.
Are you looking for a good cell phone? Yeah, why?
Sorry, I have a request. If you can move in just a little bit on the right, and also some of you in the left side, if you can just squeeze in a little.
I think this is streaming anyway.
Okay, so I'm trying to take it on the count of 3. Should we try to just point somewhere? I don't know. Never mind. We'll just take a picture. 1, 2, 3. And take one more just in case. 1, 2, 3. Thanks for supporting women in STEM. Okay, I have yet another oddball request. Does anyone have any AA batteries? Because we're not going to be able to stream without it. Okay, thanks, Asmin. So this wireless goes to the screen. So there's actually, we'd have to use this microphone that's on here. You know how to?
Does this?
Yeah. So. I don't think so. It's like totally dead. So we're gonna have to switch the mic. So you know where to get the mic Okay.
Yes. I didn't know it was.
Yeah, the batteries are dead, and I don't have extra ones. And no, we have. Okay. Well, we'll just have to stream without it. We'll try to link it with Yasmin. Somehow you'll have to link the two. I'm not sure how.
Maybe on
I don't know. We'll figure that out. Okay, can I just get, no, not your problem. Can I just get back to the screen? Alright. Okay, thank you. No, not your fault. Alright. So we are having a little bit of technical difficulties, so we can't actually stream the audio right now, so we're going to try to link the audio with the recording that Yasmin normally takes, so we'll try to figure that out later. I don't want to hold up the rest of the class, so we'll just get going. For on the final exam, just to make sure that you're aware the final exam is not cumulative. We're kind of building off of what we left off with, and we're going to continue on a little bit with this, and then we're going to transition from schizophrenia into skin. And I know it doesn't make a lot of sense right now, but there is a link, and there's a lot that we actually have to talk about before we start getting into skin cancer and then other forms of cancer. And even though we're in lecture 6, we still have a long way to go. So thank you for doing like a really great job so far. Apologies, we're just trying out today like a different note format, so we'll see how you like it or don't like it. If it gets distracting, let me know. We'll stop using them. But we are trying it out in a couple of different classes, so if you can give me some feedback, that'd be awesome. So I'm going to actually start off with a story, and this is related to your last term test. And one of the things that I want you to be aware of, that we're all here to kind of help you and support you. Your TAs are. The short answers, you're able to go and take a look at them in your tutorials this coming Thursday, so please go to tutorial, look over your term test. Your TAs are not there to take away marks if you ask for a regrade, they're there to help you. So if you go through the marking scheme, you have your test in front of you, and you have a question, but I thought that this answer was correct, your TA is not going to just randomly say, well, you're going to, if you submit it for a regrade, your mark could go down. I hate that type of response. I don't know if you've heard that before, like, oh, if you ask for a regrade, your mark may go down, or it might go up. So it won't go down, and you can always ask for a regrade. You should be free to do so in every one of your classes. But before we get there, I want you to think a little bit back to your last term test last week. And this gets into my analogy, and this is sort of what I did in research. So in research, and when I started research, out of undergrad, I felt very privileged to be able to get into research, mainly because as an undergraduate student, I didn't have the greatest grades. Like I had some A-minuses, had mainly B's, I had some B pluses, and I always felt like an inferior kind of imposter being in a lab because I just wasn't a really great undergraduate student. And so as a result of going into the lab, I tried to work and kind of overcompensate for what I perceived in my head to be these deficiencies. And so I'd be like working harder than anyone else, and I try to make sure that I was invaluable to the lab, and I tried to make myself available all the time. And as a result of that, I tried to outwork anyone else that I saw in my lab. And that, again, was mainly due to imposter syndrome. So if you've worked in a lab, and how many of you have been in a lab before, worked in a lab, research lab? Do you know what this is at the top? Autoclave tape? Have you used autoclave tape before? Do you know how autoclave tape works? No, okay, neither did I. So autoclave tape is one of the things that we use as an indicator. So we put it on anything that we want to sterilize. If we want to kill off the bacteria or anything that's growing on surgical instruments or anything that's growing inside like tissue culture media, we autoclave it. And to know that it's been sufficiently autoclaved, we put this little piece of tape on whatever we're autoclaving. Does that make sense? Seems simple enough, right? Anyways, I had been in the lab for about 3 months. I'd been working really hard trying to overcome my imposter syndrome. And I had seen like autoclave stuff coming in and then coming back out. And whenever it came out, it kind of looked like this. And I had no idea how this worked. And I thought it was kind of like, oh, well, I had my own idea about how it should be working. And so when my postdoc, who I very, very highly admired, and she was like training me, and she asked, I'm going away for vacation. During the time that I'm away, would you be comfortable like autoclaving all of the lab supplies? And because I wanted to be like really important to the lab, and because I was overcoming this imposter syndrome, I said, sure, I'd be happy to do it, not actually knowing what autoclaving entailed. And so I put the tape on, and she kind of watched me putting the tape on, and I thought, okay, this will be fine. And then I took it to the room where I knew that they were taking stuff for autoclaving. And because I was always the first person into the lab in the morning, so I got there at like 7:30 in the morning, and I usually left at 7:30 at night. I was always the first one there, first one to leave. And so I took it in, I left it, and then I picked it up the following morning. And it turns out because I was working like really, really late and coming in really early, they actually hadn't autoclaved it. And I didn't really realize that it has to be heated, it has to be sterilized under pressure, and then after the heat and pressure, the tape automatically becomes like this. It actually changes color. You can see these lines here at the top. The problem is I didn't know that. I sort of intuited that what you should be doing is actually just drawing in the lines yourself to indicate that it's been autoclaved. And so I sat there before anyone else came to the lab, and I took my black marker out of my lab pocket, and I drew in all these lines, like really, really carefully. And so this is a problem, because what happened next, because I was not communicating, I didn't actually know what I was doing, and I thought I knew what I was doing. It became a dangerous situation for our lab because a lot of our experiments became contaminated. A lot of things had to be thrown out because I had made a mistake. I thought I knew what I was doing, but I didn't communicate that I needed help, that someone had to show me what was going to make things better. And why I'm telling you all of this, as you go into your tutorials on Thursday, some of you might have done extremely well and will have done exactly what you had intended to do on term test 1. And some of you may not have done really well. And what I'm going to ask you to do is not to be like me, to ask for a little bit of help. If things did not go the way that you wanted it to go, don't pretend that everything is okay. If you simply ask for help now, everything can be okay, but I'd rather you not kind of waste your time and effort and all the hard work that you're putting in without asking for that help. Because that's what we're here for, and you can do a lot better if you simply ask, rather than make that mistake like, I think this is how it should go. And I also wanted to acknowledge my own supervisor, who always encouraged me to make sure that I'm asking questions, and that's one of the reasons why I asked you to do that, because the first thing that my research associate supervisor did, and this is Dr. Beverly Orser, she works still over at the MedSci building, and one of the things that she asks is, if you ever have any questions, always ask. and she's an anesthesiologist and the head of her department and the chair of anesthesiology, and she still asks questions. So I want you not to be embarrassed or ashamed or to feel like you're an imposter. You all belong here in this classroom, and I want you to feel comfortable enough to ask for that help whenever possible. All right, so I'm hoping that you take that message to heart, that you will make use of that. There's like no harm in asking, and if you ask for a regrade, again, we want to use it as a learning experience, and you shouldn't be afraid that marks will be taken away, etc. So if I were to take a quick poll, and I'm not going to do that for the sake of time, do you believe that exercise helps with depression? Just again, show of hands maybe if you're comfortable doing that. Exercise might help with depression. How about diet? Do you think diet is somehow related to depression or might help with depression? Okay, a lot of hands, similar hands kind of going up. And so there's a great debate going on right now. So we ended our last module with depression, and we're going to continue on that thread a little bit. I'm not going to ask you to know anything from the previous lectures, but there's a lot of debate as to whether or not diet either induces or is associated with the severity of depression, etc. And so many different studies have come out, whether it's the Mediterranean diet, the DASH diet, even the ketogenic diet. All of these different diets have been implicated as to either improving the outcome of depression or not. And it might also reduce depression risk. So if you're eating healthy to begin with, you might actually be at a lower risk of developing depression to begin with. There's so many different publications out there. How do you make sense out of all of these different types of things? So what we're going to try to do is, over the course of the next 10 or 15 minutes, is to understand how we interpret all of this different type of literature. So if you think a little bit about this, some of it intuitively kind of from a gut feeling type of experience kind of makes sense, that if you don't have a good healthy diet, that you might actually be putting yourself at risk for developing depressive symptoms. So these low quality diets, you think, would actually make you more likely to have depression. And that's that first bullet point that we're thinking about here at the top. But there's also the opposite side of things. So if you have a better diet, so you're eating like omega-3 fatty acids, you are eating much more fish in your diet, you have fresh fruit and vegetables, or you're eating things that relate to like the DASH diet, so a dietary approach to stop hypertension, all good for your overall health. Does that actually make you better in terms of your mental health and not just your physical health? And this is what the second bullet point is potentially suggesting. But there's so many contradictory pieces of literature out there. So some say yes, some say no. You probably have seen this multiple times. So some people say that drinking coffee prevents cancer. Some people say that drinking coffee causes cancer. The latest controversy is, if you eat red meat, does it protect or does it make you more susceptible to developing cancer? So how do you make sense out of all of these different forms of literature? And these different diets, they kind of make sense from a standpoint of like brain health. So having things where you're controlling for certain types of cholesterols and oils. Your brain has a lot of myelin, and therefore it needs a lot of these different types of oils, especially omega-3 fatty acids. So it makes sense that these Mediterranean diets that actually control for cholesterols and oils might be beneficial to your brain. And this DASH diet, which is mainly used to control hypertension, so high blood pressure, hypertension, good for your heart, but we also know that especially in older individuals, that if you have a bad heart, you also have a bad brain. So controlling that early might actually be a benefit too. So how do we come to a conclusion as to whether or not this is good or if this is bad? So I'm also using this as a meta teaching moment if you want to think a little bit about this. So let's take a look at this particular primary paper. So it is a primary paper. It's not the type that you should choose, but it is still considered a primary paper. If you look at this journal, the Journal of Affective Disorders, so would you want to publish in there? Sure, why not? You get a publication. All right, so would a good scientist want to publish in Journal of Affective Disorders? Is it like a top-ranked journal? Do you know any top-ranked journals? So Nature, you probably have heard that in all your biology courses. Any others? Sorry?
Lancet.
Lancet, okay, so for different clinical studies, Lancet would be a pretty highly ranked journal, has a high impact factor. You probably have heard of Science, you probably heard of Cell, and these are top ranked journals. Journal of Affective Disorders is a very, very low impact paper. Not a lot of people read it. I read it, but not a lot of people in general read the Journal of Affective Disorders, and that's why it has a lower impact. But it is kind of an interesting one because it looks at diet quality and depression risk. Those two bullet points that I just had on that previous slide, this tries to address, if you have a bad diet, are you at increased risk? And if you have a better diet, are you at less risk of developing depressive disorders? And I want us to understand how these types of studies are carried out. You don't have to worry about memorizing any of the titles or any of the other things that are on here. If you look at the boxes here to the left, it actually indicates the most important points for you directly. And again, I'll pause at certain points in this new lecture slide format to take questions if you do have any. So there's this real difficulty in establishing a link between diet as well as risk for mental health disorders. And much of this is due to the fact that when we look at the literature in general, these tend to be cross-sectional studies. And cross-sectional studies have a lot of power, but they also have a lot of hidden faults to them. So how many of you would be able to give me a really good, simple definition of a cross-sectional study? Yes?
Is it a study that looks at different people over multiple different age groups and like risk factors or whatever, and it looks at them all at one period of time?
Right, so if we are looking at just like a general population, so if I decided because we are not the same general ethnic background, we all come from different cultural backgrounds, we have different age groups within this particular room, this would be a good cross-sectional study. It's not where I'm looking at separating out whether we have people with diabetes and then control groups. This is just, hey, let's take a look and see what we find with like this general population. So this is a cross-sectional study. That's the most highly cited and highly published type of depression and diet-related study because it's the easiest. It's also the easiest type of study to look at as well. So we can't really tell a lot about, if you look at the general population, if we're looking at the third bullet point, we really can't determine, like, if we see people with depression, when did they develop it? It's hard in a cross-sectional study to do that. We don't really have a good correlation with eating patterns because they might have recently switched over, there are lots of different confounds, and so these cross-sectional studies actually can't tell us a lot about the mechanism. They're really useful, but they're not really all that powerful. For me, as a biologist, I like using it as a jumping off point, but one of the things that makes it really challenging for me to really believe that a cross-sectional study is correct is usually I want a biological mechanism. I want some understanding, like what caused this to occur. So just having a bad diet, however it's defined by that study, is one thing, but how does that bad diet actually cause an increased risk of depression? So this mechanistic understanding of that link is really important to biologists, and this goes beyond sort of just the general epidemiology where we're going into the biological underpinnings of this. So this is what we want to try to determine next. So I'm going to make a little bit of a sidestep, so we're going to come back to that. And one of the things that is, again, really quite popular right now in terms of brain diets is the supplements by something which is called nutraceuticals. Some of you might take courses with Dr. Diaz down the road, and Dr. Diaz has a course where he looks at different nutraceuticals and different forms of dietary supplements and other things. And so nutraceuticals are often one of these things that are really interesting for us because they kind of have a biology associated with them. We don't usually use them in cross-sectional studies. We use them to understand the biology. And so the next few slides are ones that I want you to have a good understanding of, and especially in terms of the highlighted, the bolded print that you see here. These are important and key points. So many of these nutraceuticals, and the one that I'm going to concentrate on is N-acetylcysteine, or NAC. Some of you might have heard it. Some of you might even be taking this as a nutraceutical, as a supplement to your regular diet, are believed to actually interfere with things like inflammation. So taking a nutraceutical might lower your inflammation. It might lower your oxidative stress. It might actually reverse any reductions in neurogenesis. If you have mitochondrial dysfunction within your cells, which leads to oxidative disruption, etc., taking NAC might actually reverse that. Now, is there a biology behind that, which is, again, for me, key? And it turns out that for N-acetylcysteine, there is indeed a strong biology that might be associated and linked with this. Strangely, and you might want to keep this in mind as well, and again, I switched away from that Journal of Affective Disorders paper. I'm just talking about a nutraceutical to understand biology right now. But one of the interesting things about this, which doesn't make a lot of sense to me biologically, is that most of these publications that deal with N-acetylcysteine, where we are upregulating things like glutathione, which is really important in oxidative stress. So if you have more glutathione, you're protected from oxidative stress. It modulates the glutamatergic types of synapses and neurons in your brain. It decreases inflammation, all these bullet points that you see up here. And it's used to treat certain types of mental health related disorders like schizophrenia. So in addition to, and this is what adjunct treatment means, in addition to the normal types of treatment that you would get, NAC is normally used to help in the treatment of schizophrenia, and we'll talk about schizophrenia in a little bit, and bipolar depression, but it's actually not used in major depressive disorders. And the mechanism, even though we think that they have a common mechanism, this last part is actually kind of interesting. We're not really sure why it doesn't work as well or why it's not as effective in major depressive disorder, but it is kind of a limitation of taking this particular nutraceutical. And you can understand why diet and dietary supplements become a bit of an issue. So I'm going to now take you through, specifically from a biological standpoint, how this nutraceutical could work, like why we think it's effective and why we think it's good for your brain health if you take it as a supplement. Is there a biology that has been well described? The biology has been incredibly well described in animal models, even in some human studies. And so the next few slides are ones where we're reasonably confident that for adjunct therapy in schizophrenia, bipolar disorder, that it works on the brain through these different mechanisms. So one thing that I hope that you kind of remember, do you remember dopamine from the last lecture? Sort of? You forgot. You did that Men in Black thing, you stepped outside, you saw the flash, you forgot everything. Okay, you have to remember a little bit of it. So dopamine is one of the neuromodulators that we're going to look at. Glutamate is a very, very common neurotransmitter. And one of the things that we know that N-acetylcysteine does, it actually enhances the release of different neurotransmitters like dopamine as well as glutamate. So if you take it, you're just going to have more of this dopamine. So you're thinking back to depression and dopamine goes down a little bit. Wouldn't this help? To me, it sounds like it would help, but the model systems that they're using actually demonstrate that in unipolar depressive disorders, it's actually not very efficacious for whatever reasons. And in addition to that, glutamate is also upregulated as well. So this neurotransmission is upregulated by the N-acetylcysteine, which is which is the sort of base model of nutraceuticals that we're going to use. Again, through these glial cells that are also part of the nervous system, so they sit beside neurons and they help to support neurons, and acetylcysteine will be converted into glutathione. And this glutathione, again, is very beneficial in general, and not only in glial cells, but this glutathione is also upregulated within neurons. And this means that in neurons, this increased glutathione is going to help to reduce oxidative stress in neurons. So think back to depression, maybe there's inflammation, oxidative stress, mitochondrial dysfunction. All of these different factors are going to be decreased by this glutathione, including like NO production is going to go down. And as a result, cell damage is also going to be decreased after taking N-acetylcysteine. So this is an important slide. The next few slides are. Before I go on, do you have any questions or things that you need some clarification around? Have any of you taken NAC as a supplement? Yes. So if we look at the NO production, so NO can also undergo very, very specific types of redox reactions, and one of those are these nitrosyl-containing reactive products, and that's going to cause cellular stress on that particular neuron. Any other questions? Is anyone taking NAC? No? Okay, no worries. So another important mechanism through which NAC might work, it primarily works, we think, and this is how I actually studied NAC sort of toward the tail end of my PhD, and one of the things that we were looking at is, does it have an impact on mitochondria within the rat brain? And we know that if your cells are in trouble, and they might be in trouble in schizophrenia, and that's one thing that we'll look at. They might be in trouble in bipolar disorder. They are in trouble in major depression, so why it doesn't work there, again, not really clear. But in schizophrenia and bipolar disorder, there's a lot of mitochondrial dysfunction. The mitochondria are just not working appropriately, and NAC actually helps to reduce sort of the mitochondrial dysfunction. In particular, if you're asking, how does it do that? It will reduce the load of calcium that is carried. The load of calcium that is carried by the mitochondria, so it reduces that. It helps to extrude the calcium, and it will also reduce the production of reactive oxygen species from the mitochondria as well. As a result of all of these different things, the cells are under less stress. There's less likelihood that they're going to undergo apoptosis. And in addition to that, through mechanisms that are still not completely understood, it increases the production and expression of a factor which is known as BCL2. I'm assuming you have all learned about BCL2. If you were to give me like a one word or an anti-something word for it, what would you call this protein? So it's an anti-apoptotic protein, and we're increasing the production of this anti-apoptotic protein here. So all of these things will help in the production of good factors for your brain, including BDNF. So when we talk about exercise and the impact of exercise on health, we'll talk about BDNF, or brain-derived neurotrophic factor. And this makes your neurons, or the stem cells in your brain, undergo more neurogenesis. You produce more neurons or neural stem cells within the brain, and it's believed to be good for brain health overall. Any questions on this before we go on? So again, make sure that you understand the different targets. If neurogenesis is increasing, this goes back to the very first slide where we looked at inflammation, neurogenesis, mitochondrial dysfunction, and each of these I'm trying to address in turn, those bolded points that I showed you earlier. Taken as a whole, if we are repairing mitochondrial dysfunction, and here we're looking at disease, like how does this occur? That part I'm not talking about. We will get back to schizophrenia and how cells misbehave in schizophrenia. But in schizophrenia, in bipolar disorder, we already start off with like mitochondrial dysfunction, reactive oxygen species being produced, oxidative stress, etc. So with this decrease in mitochondrial dysfunction directly, we will have an effect on reducing the overall inflammatory response. And we can measure that directly with our two favorite cytokines, TNF-alpha as well as interleukin-6, and we see a reduction in the production of TNF-alpha and interleukin-6. So again, another important slide. And in this particular case, I'm going to just indicate in advance that you should probably know this for your final exam. You're going to get pretty heavy coverage. I know it's about 6 to 7 weeks out, but I'd like you to start preparing as early as possible. And there's a lot of details and information on these 3 slides. And if you know that you are just not a huge fan of memorizing these things or applying these things, these last 3 slides are ones that you should probably go back, check over, make sure that you're comfortable with them. Alright, so where we've gotten to is we took a little bit of a sideline because I told you that most cross-sectional studies, most studies that we look at relating diet to mental health, normally are cross-sectional. They have a lot of confounds, they don't have a lot of biology. And then we just talked about nutraceuticals where we understand the biology. And this example of a nutraceutical would be this N-acetylcysteine. And we know that it affects how neurotransmitters get upregulated. We know that it has effects on the mitochondria to reverse the dysfunction of the mitochondria. We know that it will reduce reactive oxygen species, and it will also change and reduce those levels, and it will also change and reduce the production of interleukin-6 and TNF-alpha through reversing that mitochondrial dysfunction. So all of those bolded points that I showed you earlier is just summarized in this particular slide. So before I go on, any questions? Anything that I can clarify or things that you want to confirm at this point? Nothing? No? Okay. I'm not sure if this mic is actually working. It doesn't seem very loud. Okay, let's try it again. Alright, so if you don't have any other questions, I'm going to ask you to think back to that Journal of Affective Disorders paper. And this Journal of Affective Disorders paper actually has this overall hypothesis. Their hypothesis is that there should be some kind of association between the type of diet that you have and the incidence, how often we see depression if you have a good diet versus a bad diet. So if you were to guess, if you have a bad diet, are you more likely to have depression or not? It's a multiple choice test question. You have no idea. You're going to guess. What are you going to guess? Yes, at the back.
More likely?
More likely if you have a poor diet. Okay, that would make sense. Intuitively, it would make sense. And if you have a good diet, are you more likely to have depression or not? So you're less likely. So that would be the intuitive sort of basal response. Okay, so again, numbers not being important. This is actually based on a systematic review. I want you to have an idea of like what this actually entails. You don't have to memorize any of the numbers. You're not going to be tested on this slide at all. I just want you to have an idea of what you have to report, but you don't have to worry about putting down any of these different factors. If you go through and present like a meta-analysis paper, you should be giving these statistics, and that's sort of a different presentation-related thing. So they looked at over almost like 40,000 different articles in that very first box. So they were looking at various different digital databases. You might want to too if you're looking for different papers. So they're looking at Embase, you might look at PubMed, some of you will look at Web of Science, and they're doing like almost like a Google search where they're looking at the effective diet and the outcomes related to depressive disorders. They're also like looking at different reference lists and cross-checking them, but they start off with like almost 40,000 of these articles. After they remove any duplications, they're down to about 32,000. So what that means is you have two different databases. Some papers that you pop in from Embase or Web of Science or from PubMed, they're the same citation. So you remove any of the duplications, you've already lost about 7,000 of these. Now here's this really interesting one. Based on the criteria that they have, they remove like the vast majority. So they're looking at 32,000 articles, but they decide that based on our criteria that we're going to use, we're going to exclude about 32,000 of them. So what they're left with is 165 full-text articles, and they could have excluded it for a variety of reasons. We just can't find the paper. Have you ever done a search and you got to like this really cool paper, and then you can't actually get the paper because it's hidden behind a paywall? So they're in the same position, so if they can't get the paper, they don't include it. If they don't have, the paper's not in English, they can't include it. If the paper is not fully cited the way that they want it cited, they can't include it. If it's a clinical trial, they can't include it. So even though they start off with a huge number, they're really down to 165. And from there, they further kind of look at other criteria based on these high quality papers that they are able to access. they are excluding anything that doesn't have prospective data, so following people forward in time. If they don't have any kind of change in diet, so dietary exposure is not in their publication, if they're not mentioning the type of diet, they have to exclude those papers, etc. And so they're stuck with 29 articles. Is this a good analysis or a poor analysis? Again, guess. Good. So this is a pretty rigorous analysis, and they are including the exclusion criteria. But they started off with 40,000, and they're down to 29 articles. And based on these 29 articles, they're going to try to make a conclusion about whether diet is good or bad. So keep that in mind as we go through some of the data in the next few slides. So this is their data. So these are the 29 different publications. And each of the different publications is highlighted there for you. So the names of the authors, the year of publication, the database that they actually got this from, etc. And what they're doing here is they're comparing it to this odds ratio. So OR at the top stands for odds ratio. And what we're looking at here is this plot of whether or not you have a decreased risk of developing depressive disorder if you have a high quality diet. So if you have lots of fiber, fresh fruit, if you have sort of non-processed foods, if you have things that you would normally associate, like with the DASH diet, if you are using those high quality diets, what is the likelihood or what are the odds that you will develop a depressive disorder? And they're looking at these 29 different publications and the years of study. And so if there's no difference at all, one of the things that we would see is that each of the different publications that we see here should all line up with one, right? So in fact, what you see here is that only this publication here by McMartin et al. Is close to saying that there's no difference. If you have a high quality diet or if you don't have a high quality diet, you are at equal odds of developing depressive disorder. This other study down here at the bottom, just under it, sorry, I have like really bad eyesight. So the Akbarali et al. 2013 paper, when they looked at their data, it also showed that in comparison, they saw very, very little effect. It was negligible. There's almost no difference between having a high quality diet and developing depressive disorders. Compare that to an earlier study, or a different study by Akbarali et al. In 2013, where they saw a tremendously large effect. So they saw here a huge effect in terms of it being protective against developing depressive disorder. So first, do you understand how this data is being presented? So if there's no change, and our hypothesis is that the data from these different studies that are published show that high quality diets have an equal odds of developing depression, they would all fall pretty close to this line. Do you all see that? And this is an outlier saying that having a high quality diet is very protective against developing depressive disorder. Do you all see that? Okay, so anything on this line actually suggests that it's farther away. And so this is why you have to do this type of meta-analysis study. So many different authors using different types of individuals and across many different institutions. If you compare all their data, some say yes, some say no. But if you look at it as a whole, what you would end up with is this value down here at the bottom. And this diamond, and again, I kind of highlighted this for you. This diamond is the one that you should be looking for. And this is the summary of all of the different studies that they've looked at. So some are on this side, some are on this side, some are very close to one, some are very far from one. But if you look at all of the different publications that fit their criteria, they end up with this average down here. And this average down here says that it is 0.77 of the odds of developing depression if you have a high quality diet. So is that statistically significant? That's the key question. How many of you say yes? How many of you say no? How many of you say, I don't know, you tell me? Yes? Does anyone want me to tell them? No? No? So shall we just wait can leave that to the final exam. Tell me the answer to this question. So here, when we look at the size of this particular diamond, if it is not touching this value of 1 here, so this actually could be spread out quite a lot. And one of the things that means is that the confidence interval, the 95% confidence interval is spread out, then it would be not statistically significant. But since it's not, and since the diamond is relatively far away from this value where it would be insignificant, we can actually see that having a high quality diet is protective against developing depressive symptoms, at least in their meta-analysis. Is that good for everyone? Do you all understand that? If you were given this data, would you be able to interpret this type of data? Okay, cool, a lot of confident nods. All right, so I'll have to make it a challenging question. Are you surprised by that? Yes?
So is it fair to say you're like was it 23%, like 100 minus 0.77, like percent less likely to get depression if you have a healthy diet?
Right, so that's a great, I think your question is, if you are looking at this, how significant is that? So, I mean, it's not like you're saying that you're half as likely to develop like depressive disorder, and how significant is like 0.77. So, in terms of like looking at the statistics on a meta-analysis, this is pretty significant. It does show that having a high-quality diet is impactful and preventative in terms of depressive disorders. It doesn't give you a biological mechanism, and it just shows that there's a correlation, and it could be that individuals that have access to a high-quality diet, so let's be real, so having fresh fruit as students, like every day, might be challenging because fruit is expensive, so it just might mean that you're able to afford better healthcare, or you just have better access. We don't know, but it just does suggest better diets are protective. Even though it doesn't seem like it's like a huge effect, it's a significant effect. All right, so before we go on break, let's look at the opposite side of things. So on the opposite side of things, if you have a bad diet, does that make you more likely to develop depressive disorders? Because that's the flip side, right? We wanted to answer both questions. And this paper in the Journal of Affective Disorders wants to see both questions. And here, the data is kind of different. So if you look very carefully on these different squares, and you look at how they're all kind of put up there, here, the data is a lot noisier. So that's the first thing that you would notice with this. The sizes of the boxes actually show you how noisy the data is. So this data, for example, that you see here, is a lot noisier. There's a bigger spread, there's a bigger variation of the data than the data that you see here. It's also suggesting that the number of data points is also bigger. They probably had a bigger study that was going on, more individuals that they were looking at. But if we do the same types of things again, and we're comparing it to this odds ratio of 1, so does having an unhealthy diet make you more at risk of developing depression? And if there's no difference, it should be pretty close to 1. What their data actually suggests from their meta-analysis of all the published data that they were able to access, their value is 1.05, and there's that little diamond again. So that diamond here at the bottom, is it touching that 1 line? Yes, no, maybe? Can I get a solid maybe? Maybe, right? It's always safe. Maybe it's always safe. You can see, like even I can see, and I have poor vision, so even I can see that it's touching and it's actually crossing over. So it suggests that there's no significance. So surprisingly, a bad diet doesn't put you at greater risk of developing depressive disorder. Are you surprised by that? A little bit? And again, it was surprising for many individuals as well to see this type of diet as well. Any questions before we go on break? So for those of you who have taken other courses with me, my favorite food is quarter pounders. So I usually go out and have dinner, and either before or after dinner, I always sneak out and have like an extra quarter pounder. So quarter pounders are my thing. Sorry about that. But that's a very unhealthy diet, just to let you know. Do you have any questions, things that you want to confirm from any of the slides? Yes, a question.
So just to clarify with the number E,
Yeah.
So 1.05 means that you're 0.05 times more likely?
Yeah,
or
that's right. Which again, according to this, the odds ratio of you having a bad diet compared to a healthier diet, developing depressive symptoms is only 0.5%.
And so for the last slide,
Yeah.
it would be 0.23 times less likely? That's
Yes, that's correct. So I think that goes back to your question. And is that significant? Yes, in this study, yes, it's significant enough and would be impactful enough. Any other questions? Yes?
Is it possible that having a good diet is more closely correlated with also exercise habits than maybe having a bad diet is?
So I think that's a great question too. So is it also like correlated not only with diet but also exercise? They actually don't look at that. So you could be someone who has like an unhealthy diet but you're exercising. You could be someone who has a healthy diet and you decide that's enough, so I'm not going to go exercise. It's a good question, but these types of studies can't address that. There's no biology, there's no control, like it's literally an analysis of different cross-sectional studies. Any other questions? No, okay, so I think if you guys are okay with it, now is probably a good time to just go ahead and take our break. When we come back from break, we will try to go through schizophrenia and start to unravel schizophrenia a little bit. Yeah, come back in about 7-8 minutes. All right.
Wait, how old are you? Oh. I always forget people like
All right. If you're okay with it, I think we're just going to continue on, and I'm going to switch gears, talk now about one of those systems that I mentioned earlier, related to potentially things like nutraceuticals, potentially things like diet, and this gets into schizophrenia. And this is actually a very complex disorder, and I wanted to talk a little bit about that. Eventually, what we're going to try to do is to understand how we would approach it now in 2020 from a biological perspective. We don't have good models for many of these different disorders, including schizophrenia. What I'm going to try to do is lay the framework today for why schizophrenia is such a serious disorder. So it affects like 1 in 100 individuals. That's actually, it means that it's a very, very prevalent disorder. We don't have a good model system for it, and one suggestion has been to grow mini organoids or mini brains from skin cells and to use that to model schizophrenia and to see if some of the mechanisms that we start talking about are actually found in these mini brains that we can grow from the skin of someone who has schizophrenia. So again, it's actually being done, and there's a lot of very interesting information, and there's also a lot of controversy about growing mini brain organoids and what all of this entails. So this is where we're eventually going, and to get to that stage, I need us to all understand a little bit about schizophrenia as this disorder. So schizophrenia is one of those things that's actually incredibly scary. So you might have heard this in the news quite a lot. So I remember a few years back there was a news report of someone who was riding on a bus, and again it was shocking because at the time you didn't really hear a lot about mental illness in the Asian population, and this was an Asian individual who was sitting beside someone on the back of a Greyhound bus, and he had decided to decapitate that individual and held that bus hostage. And it made the news for a long period of time, and it's really highlighted the sort of lack of knowledge about mental illness and about how treatments could take place, etc. It was actually a shock to me personally because, again, we hadn't heard a lot about mental illness and sort of violence in the Asian community. So it was one of those things that actually drew my attention. And I'd also encountered, this was actually my first encounter ever with like mental illness. Not me directly, but my best friend in high school, and you'll understand why high school is where we often see this. My best friend in high school was an individual named Ross. And he and I hung out all the time. He's the only reason I got through high school calculus because he was like really, really good at helping me out. But I noticed, and not just me, but all of our friends with Ross, kind of noticed he started acting very, very odd. And he started to kind of act in a way that we knew something was wrong with him. But we were like 17, 18 years old, and you know, like it was just really hard for us to kind of process that, you know, he was hearing voices. He was actually acting out in a violent way that he had never done before. He started to believe that all of us were kind of conspiring against him, and that every time we were wearing blue, we were sending signals to each other that we should be attacking him in the hallways, but he'd be jumping on us before that. And it took us a while to process that he had schizophrenia. And schizophrenia, therefore, is something we don't really like to talk about. It's not something that we feel comfortable encountering. But I wanted us to understand a little bit more about this disorder. It can be inherited, so there is a genetic predisposition, but we don't have a good genetic model for all forms of schizophrenia. One of the things that makes it really hard, especially for students, even if they're undergoing treatment for schizophrenia, is that it affects cognition. You can't think really well if you have schizophrenia. So the cognitive effect is like one of the biggest sort of impairments to someone with schizophrenia. What we typically look at for schizophrenia, because it's the most scary part of it, is perception. So they see things that aren't there, or they process things that aren't actually there, or they hear things that aren't there, and so they have an altered perception, and they also have like a change in their motivation. They're just not going to act the same way, they're not going to be excited about the same things that they were before. And it usually manifests fairly late in adolescence, so high school, early university, like for males, a little bit later, mid-20s for females, but it is something that, again, affects males and females. So we don't really understand the pathogenic mechanism, so what causes this? So there are some forms of hereditable schizophrenia, but it's not like a one-to-one. So if your mom had it, your dad had it, or your dad had it, that you're not guaranteed that you're going to have it. So we're not really sure. If you look at the imaging studies for the brain of individuals with schizophrenia, it looks like there might be some changes, but it's so hard to actually process, and it might mean that there's loss of gray matter, especially within the frontal part of the brain. But again, it's not universal for everyone, so it becomes a little bit challenging. But again, most people will kind of highlight excessive loss of gray matter neurons, especially within the frontal lobe. There's also a reduction in synaptic structures on neurons, so there's a decrease in synapses, these little protrusions that connect neurons to each other. There's a reduction in synapses, and we think, and there's a lot of evidence that I'm going to present to you today, that unlike what we have in terms of depression, where we have a lowering of neurotransmission, some of these neurotransmitters actually go up. Some go down, some go up, and it becomes a real challenge. So there might be alterations in neurotransmission. We do have drugs that can be used to treat schizophrenia. They're somewhat effective in different types of schizophrenia, but they're not all equally effective for schizophrenia as well. And again, we don't know a lot about the disorder, so the treatments work in some cases, but we don't really understand 100% of why it works in these individuals, because we don't really have a good mechanism. So remember earlier, biologists, we need a mechanism because things have to make sense. So nutraceuticals, NAC, that makes sense. Why there's a correlation between having bad diet, good diet, some work, some don't, no idea, that's bad for us. So this mechanistic understanding is really important. We don't have a clear mechanistic understanding of schizophrenia, and that's one of the reasons why it's not universally effective. We can't have good therapeutic treatments for everyone that has schizophrenia, and most of the time we're just treating the symptoms. And that becomes challenging too, as I'll show you in a moment. So unlike depression, where everything kind of just goes down, you turn the thermostat all the way down, schizophrenia is different. So some things go up, some things go down. So how do you get one treatment for the brain where some things are going up and some things are going down? Really, really hard. So we'll talk about that. And we need to understand the biology. If we're going to design effective treatments, we need to know what's going on with the brain. So again, this is a slide that I'd like you to be somewhat familiar with. So you're expected to know, as the side panel shows you over here, you're expected to know some of this data if you haven't learned it in other courses yet. So schizophrenia and sort of the related disorders like schizoid type disorders. The average age of onset is about 18 in males. It's about 25 in females. And again, we don't really know if there's a prodromal period or not. There is increasing evidence now in 2020. In the last 5 years, both psychology and psychiatry and biology have suggested that even though we see schizophrenia at the age of 18 or 25, it's probably been there since the age of like 12, maybe even 14. We just don't see all of the strong symptoms until later on. So this prodromal period, and I've defined prodromal for you over there on the left, is something that we're more interested in. Because by the time we have schizophrenia, it might be too late to make effective corrections to the brain. So again, these cognitive changes, especially for individuals who are in school, and again, if you think about the statistics, 1 in 100 individuals, and think about the size of the University of Toronto, and think about the age group here at the University of Toronto, it's likely that someone has schizophrenia, that you've encountered them at some point in your university career, and those individuals will have trouble concentrating and trouble with their work at school, because their cognitive effects, the way that they can think their way through a problem, it's being affected in schizophrenia. They also have a hard time concentrating as well as being able to problem solve. So that's often one of the first things that they complain about. Just can't concentrate anymore in class. I can't really concentrate on my assignments. And so those become issues. We tend to think of some of the other symptoms as being the highlights. It's not cognition, even though if you look at it, cognition is the most important, and that's why it's the first 2 bullet points. We tend to kind of think of it more as the stereotypical suspiciousness or mistrust of others, like having those aluminum hats to block your thoughts from being read by other individuals. You might think that your TV is talking to you. So again, going back to Ross, we were at his house because his house is the only one that we were allowed to hang out with because we were kind of rowdy when we were younger. So Ross's house, we would go down and hang out in his basement where he had his TV, and the TV was off, but he would be talking to his TV because the way that he perceived things was quite a bit different. And as a result, we started to see that he did indeed withdraw from a lot of his former friends, and his mom actually was concerned enough that even though this was many, many years ago, that he got medical help because everyone was kind of worried about him. And of course, with all of that and the lack of motivation to take good care of yourself, there's usually some poor personal hygiene that goes along with all of this as well. So unfortunately, there's some really, really bad consequences if you have untreated schizophrenia. Here are some of the other things that I'd like you to make sure that you are aware of, because this will be a common theme over the next few slides as well. So if we look at schizophrenia, there are two broad things that happen to an individual. One, they actually develop stuff that they didn't have before, and that's why it's called a positive symptom. So you haven't had that before, it's been added on as a characteristic or a feature of schizophrenia. Then we have negative symptoms. They used to have this, but now it's something that they don't use as much or it's taken away. So schizophrenia has both positive symptoms and it has negative symptoms. So unlike depression, where it's usually all negative, we lose motivation, we lose appetite, we lose ability to concentrate. Here we have both positive and negative. We gain things and we lose things. Unfortunately, the things that you gain are not good. So hallucinations are incredibly common. Auditory hallucinations, so hearing voices, that's a real thing. Visual hallucinations, also a real thing. Thinking in strange ways or delusions, that's another sort of positive symptom that develops. And your inability to kind of connect rational thinking the way that you used to before. These thought disorders are very, very common. This might be the most common type. We don't, again, really know about the subcategories of schizophrenia. And in addition to that, not only are you gaining hallucinations, not only are you developing delusions, thought disorders, disordered thinking, you're also not talking as much. So you have alogia, and again, you should make sure that you know alogia is sort of a reduction or lack of speech. So you just simply talk less. So you might have been really, really chatty, but now with schizophrenia, you might not be speaking as much as you did before. And then apathy, just a lack of motivation. So it's a negative consequence of having schizophrenia. And of course, these cognitive deficits that I've pointed out several times, these are all occurring. That makes it challenging. So we have drugs that can actually treat some of these symptoms, but you actually would have to be on multiple drugs to treat all of these symptoms. There's not like one magic bullet that can fix your apathy and lack of motivation and then also make you hallucinate less, because there are different neurotransmitter systems, different regions in the brain that we think are being affected differently. And so it's a really, really interesting, very complicated disorder. Okay, so a couple of other things to mention as well. So if you were to look at most of the literature, not all, but most of the literature would suggest that at some point there's an underlying change in dopamine neurotransmission. So some of the features that I'll get to again in a moment, there's some kind of increase in dopamine neurotransmission within your brain. So again, really interesting, and over the last sort of 50, 60 years, that's been the major driving hypothesis to account for having hallucinations, delusions, etc. It might be because you just have too much dopamine around in your brain. And we'll talk a little bit about that. And in fact, if you have someone who is on a psychostimulant like amphetamine, which definitely overactivates the dopaminergic system, there's just too much dopamine around, and that's why you have, the effects of amphetamine work the way that they do. If you have a chronic user and you are someone who continually uses amphetamines, they actually develop psychoses. So again, the way that science works, if amphetamine overactivates the dopamine system, and then as a result, these individuals that have been using amphetamines now develop psychoses, these might be linked as the positive symptom in schizophrenia. Does that make sense, that line of reasoning? Kind of, that's science, I guess. And again, these psychostimulants can actually produce paranoid psychoses, even in healthy individuals. And if you have schizophrenia, taking amphetamines actually make the hallucinations and psychoses, the inappropriate thinking, disordered thinking, even worse in these individuals. So it suggests that dopamine is important in schizophrenia. Again, it's kind of interesting leaps of logic here, and then the science kind of goes in and tries to fill in some of the gaps. And that's where we're kind of going here today. So I just read a really cool paper on schizophrenia. Do you know of any one thing that can actually protect you against developing schizophrenia? Any one thing. I don't think you can actually Google it. Unless you have access to my Google browser, which you shouldn't have, by the way. If you do, don't look at it. Any ideas? Yes?
Well, I mean, just a lack of mind-altering drugs.
A lack of drugs? Sure, could. But like one thing that, even if your mom, dad, genetically predisposed, family history of different mental illnesses, one factor that actually will protect you 100% from developing schizophrenia. Anything? Yes?
Does that have something to do with like geographic location?
Nope, not geographic location. Good guess, yes?
Do you already have something that's like messing with your dopamine system and you can't reduce it?
Already have something messing with your dopamine system? Nope. You can have stuff messing with your dopamine system, like depression, you can still develop schizophrenia. Anything else? Sleep? Okay, not sure what that means.
You sleep more.
If you sleep more, you're protected from schizophrenia. I wish that were true, not the case. Yes?
Increase of omega-3s.
Increase of omega-3s. Diet is actually not going to protect you from schizophrenia in this particular case. Nope, not 100% protected. Yes?
Or good social support?
Good social support. Okay, maybe, possible, but not always, no. Okay, I would like to think I was a really good friend to Ross, but he's still. So just saying, like anecdotal, but not always true. Yes? Exercise. Again, kind of the jury's out on that, but it's not 100% protected. And again, I'll talk maybe about that really briefly in the next lecture. It's if you are born blind, and you laugh, but it's actually really counterintuitive. If you're born blind, you're 100% protected from ever developing schizophrenia. Even if your family history has schizophrenics in your genetic background, you're 100% protected. Here's the mind-blowing part to that though. So I just read this paper before coming to class. The mind-blowing part to this is, if I blindfold you for 3 days straight, you actually develop visual hallucinations. So there's a link between vision, hallucination, psychoses, but if you don't have vision, you're protected from developing that. And it's completely amazing. So I'm kind of reading through that paper and trying to follow up on some of those papers, and I'll try to bring that after reading week so we can look at that together. So completely amazing paper. Let me take you through this slide. There are some important things on this slide. You don't have to memorize all of it, and as the bullet points over there on the left pane tell you, you should know some of the different aspects of the neuromodulatory system. So technically dopamine is not a neurotransmitter, it's called a neuromodulator. So it modulates the transmission of other ions, but you can use it interchangeably, I don't really care, but that's why I have it as a neuromodulator. I want you to be aware of these two major areas that I'm going to be pointing out to you in a second. So one of these areas that produces dopamine is known as the ventral tegmental area. So this group of neurons, this blob here in blue that the laser pointer is on currently, this blob of neurons, there's a group of neurons, their cell bodies are there, and then they send out their axons, which is what you're seeing here. And the axons at their ends will release dopamine. All the cell bodies that release dopamine begin here in the ventral tegmental area. And they release dopamine. And they will release dopamine onto a number of different structures. And these structures include the nucleus accumbens. So that structure you should know is a target for dopamine release. They will also release onto the amygdala and the hippocampus potentially, and then all throughout the frontal cortex. And beyond that, you don't really need to know anything else. The other area that I want you to be aware of is this area here within the midbrain. So within the midbrain, we have this other group of neurons, which is the substantia nigra, which I'm sure all of you, if you've ever taken physiology courses, if we lose substantia nigra neurons that normally produce and release dopamine, what disease results? What disease results? Yes. Parkinson's, right? So you've all heard of Parkinson's disease. And normally they also make synaptic connections to a variety of other midbrain structures and cortical structures as well. But I just wanted you to be aware of these two midbrain related, because they're both found in the midbrain. So they're both found in this area. And they both are producing and then releasing dopamine. And they have different types of projections. And so, because the substantia nigra is involved in controlling motor functions, this is where motor pathways could be affected. And this pathway between the ventral tegmental area and the nucleus accumbens is thought to be involved in addictive behaviors. And if you have like too much dopamine release, it might lead to addiction. But because of the way that dopamine spreads throughout the entire brain, their axons reach like far distances throughout the different parts of the cortex, we know that this actually has far-ranging effects. Not just the midbrain, but the entire brain is affected by dopamine under normal circumstances. Do you know what L-DOPA is? Again, going back to physiology, if you knew about Parkinson's disease, what is L-DOPA? So if you are missing dopamine, you can give L-DOPA therapy and it gets into the brain and then gets converted into dopamine, right? So you've heard of L-DOPA? Do you know what the side effect of taking too much L-DOPA is? Outside of the movement dyskinesia, it's like outside of the movement part of it, the tardive dyskinesia is, what else happens if you have too much dopamine? So dopamine, if you have too much of it, through L-DOPA, you can actually have psychoses, hallucinations, and you can also alter your thinking as well. So those are well documented. So having too much dopamine has many of the same effects on Parkinson's patients as we would see in schizophrenia. So they see the same sorts of changes that are going on there. So again, I'm going to talk to you a little bit more about dopamine. I don't want this to be a neuro lecture, and I just wanted to talk to you a little bit about what we know about dopamine. In some parts you need to know, in some parts you don't need to know. So I'm going to actually begin on the left and talk to you about what's happening over here at this particular synapse. You don't need to memorize all of the different parts to what's here on the presynaptic element. So what we're looking at here is the synthetic pathway to make dopamine. So how do we make dopamine? So it starts off with the amino acid L-tyrosine. So you have to take that in through your diet. You eat different proteins. They get broken down to amino acids. Some of those amino acids are like L-tyrosine. Then through this enzyme, which is known as TH, and TH is tyrosine hydroxylase. And apologies, I don't think I wrote that down for you. Tyrosine hydroxylase converts L-tyrosine into the first step in making dopamine, which is L-DOPA, that compound that you can buy as a pill, that you swallow, take, and it makes more dopamine. And in fact, there is that dopamine gets packaged into neurotransmitter vesicles, this little oval that you see here. And then under the appropriate conditions, it gets released from the synapse and then reacts with receptors on the next neuron over. So the synthetic pathway is one I'd like you to make sure that you know, mainly because of what I just glossed over here on the right. Dopamine has a number of different receptors throughout the nervous system. One of these is known as the D2 autoreceptor. And if you look, where do we find the D2 autoreceptor? So we call this compartment the presynaptic compartment, and we call this compartment the postsynaptic compartment. Where is the D2 autoreceptor normally found? Presynaptic. So again, I'd like you to make sure that you are aware of it. If you've learned it in other courses, great, so you don't have to relearn it. If it's new to you, make sure that you know that D2 autoreceptor is here, and it also responds and acts through dopamine. It triggers a G protein coupled pathway, and it actually causes the activation of this pathway to make more of the dopamine. Okay, so just for the time being, ignore this sort of inhibitory arrow coming back. So normally it's sort of a positive feedback loop. We need to block this receptor, which is why I have this negative feedback loop here. But it normally causes more dopamine to form if it gets continually activated. So just keep in mind this D2 autoreceptor. Once you turn it on, if you're not blocking it, it just continually forms and pumps out more of this dopamine. So studies in a lot of populations, again, suggest that there's a link between amphetamines and amphetamines that you see here. They normally block the reuptake of dopamine. So if you stop the reuptake of dopamine, it means that you have a lot more at the terminal. And that means the dopamine's there for a longer period of time. And if you have too much dopamine through blocking the reuptake of dopamine, that means that you will develop psychoses, and we link psychoses with schizophrenia, and therefore since the outcomes look similar, schizophrenia must be caused by too much dopamine. That's the sort of logic that science has been using over the last 50 to 60 years. So what I tried to do is to introduce you a little bit to dopamine, a little bit to some of the theories as to why we think too much dopamine causes psychoses, which is one of the side effects that we see within schizophrenia. So again, it's equaled to schizophrenia by most scientists, at least over the last 50, 60 years. Some scientists have started to rethink that theory, but this is still pretty dogmatic, or part of our normal thinking, or part of our normal model for the system. And it's not just exclusive to schizophrenia. So if you elevate dopamine, not just in schizophrenia, but if you elevate dopamine through taking L-Dopa, you develop a psychosis. If you elevate dopamine through taking drugs like amphetamines, you also develop psychoses, especially if you take it over a long period of time. But maybe it does underlie what we see in schizophrenia. So this is that current model. I'm going to try to take you through like how did we make this logical leap, and why are we equating too much dopamine with the psychoses with schizophrenia. Just to make sure that you're all on the same page, what two types of major classifications of symptoms do we see in schizophrenia? So we see, yes? Positive and negative, and psychosis is a positive or a negative symptom? Positive symptom. So we normally don't have it, it's added on, so it's a positive symptom. So most of this actually comes from studies that were done in the 1950s. In the 1950s, there was this huge explosion in pharmacology and toxicology studies because the rat model started to dominate, and this rat model became a model for human studies. And so as different drugs were being developed, one of the first drugs that was developed that was found to be safe in rats and mice was this drug right here in the middle, haloperidol. And haloperidol was found to be, in different animal model studies, to be found safe at relatively high doses. And so they decided to use this in clinical trials in schizophrenics because it was found to bind to dopamine receptors, and it was shown to be a very strong antipsychotic. It could reduce the psychoses, in other words. So haloperidol became sort of this model system by which, since this one drug, we think, is binding to dopamine receptors and blocking dopamine signaling somehow, that there must be too much dopamine, fitting in with all of the other things that we now know about amphetamines and L-Dopa, too much dopamine around, and the fact that you can block dopamine receptors with haloperidol and it becomes antipsychotic, then this fit in with, this is how we derive this model. So this must be the way that this works. If this drug reverses psychoses, and it's also working on dopamine receptors, and in addition we have other things that cause dopamine to be overactive and it causes psychoses, it must all be related to dopamine. Is that a logical conclusion? Might be. Is it something that you understand how they got to? Maybe, right? So that's sort of where science is kind of at. And in fact, if you go back and take a look, one of the things that actually is really, really interesting about this is you can inject in a radioactive tracer. So here we're using the more powerful radioisotope, the 11 carbon, so it emits much more vigorously, so we get a better signal that is labeled onto raclopride. So think of this as being, where is the most dopamine signaling going on? This is in the area of the midbrain, so just to orient you a little bit. And if we register this on an MRI scan, if we have an untreated schizophrenic individual, there is a lot of dopamine that we see there. So raclopride is tagging all the dopamine signaling areas. There's a lot. So raclopride is definitely showing our theory, at least, is somewhat correct. And then if you go in, haloperidol is that drug that I showed you has antipsychotic effects. Does it actually work by reducing dopamine signaling? And we go back in with the raclopride after treatment. We show that, yeah, when they start to reduce their psychotic behavior and they no longer have psychoses, we also see a reduction in their dopamine signaling. So again, it doesn't it's not counteracting our theory about dopamine being the major driver for psychoses in schizophrenia. So again, overactivity of the dopaminergic system is thought to be really important in schizophrenia. It's also thought that there might be too much activation of the D2, the autoreceptor that I showed you presynaptically. If we can block that D2 autoreceptor, we can block the schizophrenia, and that's how haloperidol is thought to work. It binds to the D2 receptor, it inhibits the activity of tyrosine hydroxylase. We don't make as much dopamine anymore, and that's why we saw the reduction in dopamine signaling. Since that's important, and I want you to make sure that you know that for your final exam, are you all clear on that? Do you need me to repeat that, or you want to confirm anything, clarify anything? The D2 autoreceptor normally causes more dopamine release. Here, when we block it, it inhibits tyrosine hydroxylase, TH, and so we form less dopamine, which fits into what we saw here, less dopamine sort of at this particular individual. And again, it's an interesting theory, and sorry, I have to also go back. I don't know if you're aware of this, we also have signaling of dopamine in other disorders as well. So it's not just in schizophrenia, it's not just a reduction in Parkinson's, but we also have changes in dopamine signaling in detachment disorder, as well as in Huntington's disease. In fact, in both of these disorders, there's just way too much dopamine around as well. And we can use the same sorts of treatments there. Yes, you have a question, comment, clarification? Yeah.
Just from what I had learned in the past, so D2 was from my understanding, it was blocked on the postsynaptic cell, and that was how it blocked dopamine binding, and that the D2 autoreceptor actually did inhibit tyrosine hydroxylase. So that's not the case.
So
it's the same receptor as what my understanding was, but on pre and postsynaptic cells.
So the pre and postsynaptic actually connect with different types of G protein signaling complexes. We're just going to keep it somewhat simple here because we're not going to go into the background neurobiology. And we'll just stick with sort of the reduction in the overall dopamine signaling. There are like other interesting and other theories around dopamine signaling and how this could be mediated through haloperidol or other D2 blockers, but we'll just keep it somewhat simple for the time being. But it's a great question. Any other questions that I can clarify for you now? No? Okay. Right. So somehow this dopamine signaling is being dysregulated in schizophrenia, at least if our data supports this model. And this means that we have to understand a little bit of the physiology of what dopamine normally does. So why do we need it, and where is it normally useful? And so when dopamine neurons or dopaminergic neurons normally fire, we get the strongest activation. Yes, sorry, I didn't see your hand.
Yeah, I just have a question about the last one. So just the D2 receptors, when activated, naturally inhibit tyrosine hydroxylase, correct? Like based on the slide?
So I'm talking about it being inhibited through the actions of like haloperidol to prevent more dopamine being released and more dopamine being formed.
Right, so is haloperidol like an antagonist?
It is an antagonist and it's a blocker for the D2 receptor. I'm looking at it primarily through a presynaptic mechanism. There are some theories that it works on a postsynaptic mechanism where it's just decreasing the effectiveness of dopamine signaling postsynaptically. But I'm looking at it in terms of the overall levels of dopamine that are being produced, that are being blocked at a synthetic level. Yes, Omar.
What did QED stand for?
QED.
On the previous slide.
I think that's a mathematical term. I think. It just means that therefore it is, or something like that. Proof of concept. Sorry, I'm not sure what QED actually stands for, but I think that's the mathematical term. That just means that therefore. Okay. Okay, so, all right, so when we get a novel stimulus, so if something is like capturing your attention, it's something you haven't seen before, something new and dynamic, we get a little burst of dopamine that's getting released. And that's good because it makes us pay a little bit more attention, makes us more aware of like what makes it unique. And that is normally how we think it works. It might also signal for a reward in uncertain environments. So getting a surprise, some of you like it, some of you don't like it. I guess we'll find out on the final exam, maybe. Or if you know that a reward is going to come, then maybe having dopamine preparing you for that reward is also a use as a signaling molecule. And then it also might be a signaling molecule for how to avoid certain things, or making an association. And this gets into a classic experiment that I'd like you to make sure that you know from Wolfram Schultz, which I'm going to take you through next. So in the 1970s, there were a number of different researchers that were interested in Parkinson's. So Parkinson's was like the dominant field within psychology and neuroscience back in the 1970s. They used to do primate animal studies as their primary model system, not rodents. And they wanted to understand the mechanisms by which Parkinson's disease kind of came about, how to find therapeutic approaches to it. And one of the interesting things for this was that they knew that the dopaminergic system was involved. It's not until the 1980s where we understand dopaminergic neurons are being lost through oxidative damage or potentially like toxins in the environment like MPTP or other things. But in the 1970s, they knew dopamine was involved, they knew that dopamine was important, and they wanted to know how these neurons that release dopamine, some of which we've covered, like the substantia nigra, how they might actually be involved in movement control. So in one of these classic experiments, Wolfram Schultz's lab in the midbrain, so they're kind of placing their electrodes within the midbrain, they missed. They were trying to find a small group of neurons, they're putting in these stimulating electrodes to electrically stimulate these dopaminergic neurons. And if you go back to an earlier slide about how close they are at the level of the midbrain, we know that the substantia nigra and other dopaminergic neurons, like the ventral tegmental area, are relatively close together, and he kind of missed. And one of the things that he didn't know is that he was actually in a different area of the midbrain, not the substantia nigra, dopaminergic, but these were more involved in reward behavior. So they release dopamine, but they're involved in reward, which is now what we classically know. And so what he was able to show is that there are certain groups of neurons that actually respond to food rewards. So even though he had missed and he hadn't found the neurons that control the movement of the arm, he was able to show through like some really elegant experiments. So this is, again, if I remember correctly, so he had been trying to find a neuron that controlled like the movement of the arm. He missed that particular dopaminergic neuron that controlled arm movement, but every time the neuron that he was recording from, whenever the animal that he was recording from saw someone picking up food, it got really, really activated. And then he realized that, oh, I missed the movement control neuron, but what I'm looking at is sort of a reward representation neuron. And so he decided to test out this system, this dopaminergic system, and he wanted to understand what dopamine is normally doing in this particular context. And I think this is my last series of slides, and let me take you through this really, really quickly. It's a classic experiment, so hopefully you've maybe heard different elements of this before. So here's this recording. He's recording from a reward neuron. This reward neuron responds to food, so it likes knowing that there's going to be food. So here's this first recording that he does from this neuron. So this neuron is just firing away normally. So you see each of these spikes that you have up here are actually the cumulative activity of each of the different individual recordings. So each time you see a little dot here down this axis, if we pile them all up, we get this little spike that results. It just shows you that the neuron is active, becomes a little bit less active over here, so we get a little dip. Oops. And then it fires again a little bit, so we can see more activity. And then, when we're recording from this dopaminergic neuron, Wolfram Schultz's lab gives the animal food reward. So this is where we see the line, we see the R, that's that food reward. That previous slide we were measuring from a dopaminergic neuron that has food representation as a reward. And then what do you notice about the activity of this dopaminergic neuron after the food reward is given? Nothing. Do you notice anything? Yes.
The activity spikes.
Right, so we see much more activity in this dopaminergic neuron. It is responding to this food. And then it kind of goes back to baseline. So first, you all see that. Are we good? Kind of good? Do you need me to go through any of this again? No? Okay. So he does this other really cool experiment. So, you might argue, and I guess it kind of is counterintuitive now, but you might argue, okay, is the dopaminergic neuron actually firing because of the food reward, and just the food, or is it something else? And so he does some other experiments which are really cool, goes back to Pavlovian conditioning, and he does a conditioned stimulus. So what he does is, during the time that he is doing this experiment again, instead of actually giving the food, he trains the monkey or the animal first, the primate first, and he conditions that every time a bell is ringing, a few seconds later you're going to get a food reward, and then he does the recording. So he trains, you hear a bell, you're going to get a food reward in a few seconds. Is that clear for everyone? Second experiment. So what he does here is he has done this conditioning, so this is done before. So he's recording baseline, 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 a little bit of activity. Then he actually gives the conditioned stimulus. Again, I'm not sure if it was a bell or if it was something else that was going on. And then he does the conditioned stimulus. What do you notice happens with the dopaminergic neuron? After the conditioned stimulus, get more dopaminergic activity, which is what you see here. And then a few seconds later, as promised, the animal gets the food reward. What do you see happening with the dopaminergic neuron? Nothing. So it's not responding to the food per se, it's responding to the anticipation of the reward. Is that clear for everyone? I'm hoping that's okay for everyone. So again, that was a really cool experiment. That was telling us a lot about what dopamine is doing. So dopamine is firing not because you actually got something to eat, but I'm like excited because I might go out to eat later on, and that gets my dopamine going. In addition to that, he does this other really cool experiment too. So what if you are a total jackass and you decide not to give the animal a food reward? What happens to the dopamine? And it also has some really important implications as well. So this last experiment is where dopamine is now thought not only to be able to predict, like, oh, I'm going to get a good food reward, dopamine gets a little spike, but it also tells us that dopamine can go completely silent when what we anticipated actually doesn't happen. And so here, the conditioned stimulus, ring a bell, the animal spikes the dopaminergic activity, same as we saw in the second panel. Then it goes back down to baseline. Normally it's supposed to get a food reward. It doesn't get a food reward. And then what happens to the dopaminergic neuron? It goes silent, correct? And so this difference between firing as well as going silent allows dopaminergic neurons to tell you when something is rewarding or when something that you predicted didn't happen, and it gives you some idea of rewarding versus like prediction of like errors. And so this is where we're sitting right now with dopamine. So if dopamine signaling is going awry, and this is what dopamine is normally doing, telling you when something is relevant, important, and what should be happening next, this model becomes like a really important one for us to examine. So this is why I guess we should never promise you something and then not follow through with that promise, right? It becomes like a really bad sort of dopaminergic error prediction model. So I think at this point, I'm just going to remind you about tutorials this week. You get to go to your tutorials, you get to go through your term tasks. If you can bring like a printout of the first page of the paper that you might consider presenting, and then try to form groups as early as you possibly can. You can always change groups, you can always change papers, but getting an early start is useful. Please go through your term papers with your TAs. If you ask for a regrade, they will do so for you. Other than that, please take care of yourselves during reading week. Have a great, restful reading week. See you all in 2 weeks' time, and have a good break if you are taking one. We're really going to need your recording today for sure, because we're going to have to try to...